Yes, ma'am.
Okay, you can start presenting your lecture.
Yeah, ma'am. Dr. Kalyan Lakshmi is going to start.
Hello?
Yes, ma'am. Can you hear me?
Yes, yes.
Dr. Kalyan Lakshmi is starting, madam, and I'll
Okay,
follow her.
okay, okay.
Yeah. Ma'am, madam's audio is not working. She's trying something else.
Hello, ma'am. Hello, ma'am. Can you hear me?
Yeah,
Yes, yes.
now you're
You can
audible.
proceed.
Are you sharing PPT, Kalyan Lakshmi?
Hello, ma'am.
Yeah, you're audible.
Ma'am, okay. Just okay. Good afternoon. Myself, Dr. Kalyan Lakshmi. Our project is on green synthesis, metal smart nanoparticles in breast cancer therapy and drug delivery. Our task is 82 and 83, and another is magnetic oxide nanoparticles for targeted drug delivery and magnetic hyperthermia. And co-PIs are Professor P. Sandipta from Department of Microbiology, Usman University, and myself, Dr. Y. Kalyan Lakshmi from Department of Physics, OU. And our basically the research is on the cancer cells. So cancer is one of the leading causes of death worldwide. Among 20 million new cancer cases, nearly 10 million cancer deaths occurs globally every year. So in 1 in 5 people develop this cancer during their lifetime. There are many reasons for expected due to the lifestyles, pollution, environmental exposures, tobacco, alcohol, and lifestyle, obesity, etc. And the different types of cancers which exist are the breast cancer, particularly seen in women, and the lung cancer in both the genders, and prostate cancer in men, and cervical cancers in women. So some of the conventional treatment methods, which includes chemotherapy, radiotherapy, surgery, and immunotherapy. And there are several disadvantages with reference to the above conventional methods. Some major problems with these treatments are non-specific targeting which damages healthy tissues, and there is a severe side effects such as hair loss, fatigue, organ toxicity, etc. And due to this drug repeated giving drug to the human body, it accumulates at the tumor sites, and there is a multi drug resistance in cancer cells, and the high dosage reduces treatment efficacy. Tumor recurrence and metastasis remains a major challenge. So there is a need for another advanced.
Dr. Kalyan Lakshmi, sorry to interrupt. Your slides are not scrolling.
No, ma'am, I am still in the same slide.
Same slide? Okay, sorry.
You're not able to see? Kanpinchadleda?
Yeah, I can see it.
Ma'am, can you see the slides?
Yeah, now I can see. Yeah, I think you have to go
Now
into
can you see,
slideshow
ma'am?
mode. It is just in
I have
PPT.
changed the
You
slide.
have to
Oh, slides are there, ma'am. You go to the slide that
slideshow mode.
slideshow mode.
I'm going to the slideshow only.
You have to play
Ma'am,
the
can you see the
slideshow.
slide now?
Yeah. Now it's
Magnetic hyperthermia is
this
one
is better.
such magnetic hyperthermia is one such treatment, and it has several advantages. And what actually magnetic hyperthermia means is it is one of the best cancer treatment techniques in which the magnetic nanoparticles are introduced at the tumor tissue site, and it can be heated locally using a magnetic alternating magnetic field. So this alternating magnetic field generally raises the temperature to about 42 to 42 to 46 degrees centigrade, and this will destroy the cancer cells. And due to this treatment, there is a minimum harm to the healthy tissue. Only it targets only the cancer cell. So how this heat is generated? In generally, the magnetic nanoparticles due to this magnetic spin which it possesses. So by applying the alternating magnetic field, there is there is a change in the spin orientation all the time. So there is a Neal relaxation and Brownian relaxations occurs, and which due to this a localized heating is created at the cancer cell site. So due to this, there is means if the temperature is maintained between 42 to 46 degrees, then these cancer tumor means the cancer cells can be destroyed at the particular site. So this is one of the best techniques which is even approved by FDA. And why particularly iron oxide nanoparticles? As we all know that iron is a one such good material where it possesses good magnetic behavior. So particularly why only nanoparticles here is nanoparticles possess unique physical and chemical properties due to its high surface to volume ratios, and in which so it can reach up to the cell size, so that this magnetic, by applying the magnetic field, alternating magnetic field, this, as we have discussed, it can be reduced. And not only that, it is also biocompatible, because it is, we are dealing with the human cell. So obviously the material should be compatible with the human cell, and also biodegradable. So iron nanoparticles with particularly magnetite and hematite phase are favored in medical applications due to biodegradability, so that the body can metabolize the iron through normal biochemical pathways. And from the literature, we have found it has been found that nanoparticles less than 100 nanometers sizes particularly exhibit super paramagnetism, and this super paramagnetism is nothing but which has got zero remnants. So therefore the magnetic nanoparticles less than 100 nanometers are required for this treatment. um, that is the even the FDA also approved the nanoparticles, iron based nanoparticles for the clinical use. So therefore the study will be continued in the iron based nanoparticles. Why the magnetic response? It has good magnetic response particularly, and you can modify its surface easily, and you can control the target drug delivery, and heat generation can be undertaken at the alternating magnetic fields. And the objectives of our present study are to develop stable and biocompatible iron oxide nanoparticles targeting using ligands and antibodies, and to evaluate therapeutic efficacy via drug delivery and hypothermia. So studies here we have, and three of us are working in this project. One is myself, Dr. Kalyan Lakshmi from Department of Physics, that is from the Spoke University. And the role from our department is the synthesis of magnetic nanoparticles and the study of their structural and magnetic characterization, particularly both in AC and DC magnetic fields, how it responds and what temperature can be attained with the magnetic field, tuning of the magnetic field and the temperature in the studied prepared nanoparticles. And Dr. Sandeepta ma'am from the Department of Microbiology will be investigating on the analysis of biocompatibility of iron oxide nanoparticles in vitro and in vivo. And from the Hub University, Dr. Venkataya will also be working on the synthesis of some more magnetic based nanoparticles. And as we do not have some of the studies, and the magnetic behavior will be completely studied over there. And in the first few 6 months, we have prepared Fe2O3 nanoparticles using a sol-gel synthesis technique, where we have taken iron based salts, and which is dissolved in the solvents, and then citric acid is added to it, and thereafter slowly heated to form the gel. And finally we have got it is automatically, means auto burnt, and we have collected the powder and just calcined it for at very 500 degrees centigrade, and finally dried it such that we got the nanoparticles here. In order to determine whether any other impurities are present in the material, and it has got the hematite phase, the XRD has been carried out, XRD measurements were carried out, and here it forms a good crystalline nano-sized particles are formed here. The diffraction peaks were indexed to the peaks pertaining to alpha Fe2O3 is present here, which belongs to the rhombohedral structure. So we have confirmed the phase, and it possesses alpha hematite phase here. And next is the bondings, iron oxide, because it is we have to again coat it with the polymer and therefore FTIR studies were also confirmed, and it possesses both iron oxide based bondings. One shows at 540 centimeter inverse and another at 460 centimeters inverse. This is what we have studied. And the magnetic response is also found, the magnetic versus temperature, because as these materials require room temperature magnetization. So to find out the magnetization here, as we have DC magnetization right now, is to find out its magnetic behavior at room temperature. Here we have carried out magnetic field versus magnetization and possess ferromagnetic nature. This response we have founded from the magnetization. So up to this point we have done, and the further work on these nanoparticles will be further carried out in the microbiology department. And I request Sandeepta ma'am to continue the presentation.
Thank you, Dr. Kalyan Lakshmi. Can you stop sharing your slideshows?
Yeah.
I'll share. I hope my slideshow is visible.
Ma'am, is it okay?
Is it visible?
Hi,
Yeah, is my
yes.
slide visible? Yeah.
Slide is
Okay,
visible.
Yeah, thank you. Thank you, Dr. Kalyan Lakshmi, and I thank the organizers,
Yeah, it is
Professor
visible.
Vijlata madam. Yeah, I'm continuing, ma'am. Thank you.
Yes.
Yeah. So thank you, ma'am. So as Dr. Lakshmi has explained about the synthesis of magnetic nanoparticles, so main thing we are trying to do here in microbiology is to design a tumor-targeted nanoparticle delivery system. As you know, there are several chemotherapeutic drugs which are available, and even immunotherapeutic drugs, they have a very big role nowadays in treatment of cancer. So as Madam has told about magnetic nanoparticles, which have a very good property of hyperthermia, which can heat the surrounding tissue. So when these nanoparticles are applied to the are injected into the tumor, and alternating magnetic field is applied to this area. So there are temperature increases up to 113 degree Fahrenheit. So what that does is it destroys the surrounding cells and tissue and even blood vessels. And this helps in destruction of tumor cells. So in our project, what we want to do is carry out drug loading onto these nanoparticles, because they are already having a very good hyperthermia property. So we will functionalize the nanoparticles with a targeted ligand like doxorubicin, which is a generic chemotherapeutic drug, or antibodies, and we want to test this in vitro to see if it is having any synergistic effect and specific killing or specific effect on cancer cells, specifically in breast cancer cells. And why we want to use this is this helps in targeted delivery, which is the recent development in cancer therapy, because previously conventional drugs had a very low rate of tumor localization, very high systemic toxicity, and they had very specific role in killing cancer cells. But if we can functionalize these traditional therapeutic drugs with iron oxide nanoparticles, this tumor localization can improve, because it will be directly injected into tumor, and also be added with a homing agent, which is a therapeutic drug. It will be having multiple functions, because it can be used as a drug, it can be used in diagnostics, that is called as theranostics, as Professor Haripadma Sri Madam also talked today. And also there will be very responsive stimulus because of the homing agent, which is attached to the iron oxide nanoparticles. So these nanoparticles will be coated with homing drugs using a linker, specifically dextran coating, as you can see in the figure, or polyethylene glycol chains. What they do is they help in better stability of the targeted drug, which is attached to the nanoparticles where there is iron oxide code. And they provide very highly controlled drug delivery. So in this, we will carry out drug loading, then we will characterize the loaded nanoparticles by various physicochemical characterization techniques like XRD, TEM, zeta potential, VSM, and so on. And once these are coated, we will be using these nanoparticles in vitro first, where we will check using magnetic hyperthermia to see if it is specifically killing cancer cells or not, and not killing normal cells. So as I said, dextran coating is one of the central properties of this work because it is known that it not only prevents oxidation but also helps in enhancing multifunctional sites for attaching therapeutic ligands onto the nanoparticles. And it improves colloidal stability in biological media and also reduces opsonization and immune response, which may be difficult, which may cause difficulties in general chemotherapeutic treatments. So recently we have used dextran to coat onto this nanoparticles. Himabindu, who is the PhD scholar working under this Pair grant, she is at present functionalizing the nanoparticles provided by Santosh at Dr. Kalyan Lakshmi's lab, who has joined the grant. And we just conducted FTIR and we have confirmed the successful attachment of dextran to the iron oxide surface. So this is the data we got recently. And now we are going to use this dextran coated nanoparticles to functionalize using different drugs like presently we have plant extracts which we have previously studied showing very good toxicity against cancer drugs. So we'll be using those, and even the recent drugs which have been used like doxorubicin, we want to load the nanoparticles and test their efficiency in cancer killing, and also kinetics will be studied in this area. So usually for cell culture, in vitro studies are done using MTT assay and scratch assay or cell invasion assays, clonogenic inhibition assays. And since we are using magnetic nanoparticles, we'll be studying hyperthermia generation also. And we will also test if this effect is specific to cancer cells by testing hemolysis and protein corona properties, which will help in estimating the biocompatibility of these nanoparticles. So this is the overview, and finally the drug-loaded nanoparticles having this iron oxide core will be tested in various ways to test if they have improved apoptotic effect, better magnetic hyperthermia effect, and cellular uptake, and so on. And once these studies are completed, we will be testing the nanoparticles on a small animal model, namely mice. We can use nude mice in this, which have no immune response. So the tumor cell stability is better in these mice. They are not easily destroyed, unlike wild mice or biopsy mice or black mice, which can easily eliminate the tumor once injected. So we can use this model, and we can also use the biopsy model in some cases. So we will test different models by testing the alternating magnetic field, which is going to generate the hyperthermia. And once the drug-loaded nanoparticles are injected into the tumor site, definitely there should be hyperthermia generated, but we will test if the drug-loaded nanoparticles have better efficacy. The current issues in this magnetic hyperthermia, which is used in thermal ablation of tumors, is that they are used for small tumor sizes like 1 to 2 centimeters. So larger tumors inevitably are surgically excised, in which a large part of the organ is also removed, which has a lot of the concerns among patients. So we want to see if our intervention can help in targeting larger tumors. And recent developments in anti-tumor drugs, if you can see on the slide, these are some drugs like ivermectin, fenbendazole, and mebendazole. These are creating a storm in anti-cancer therapy. These are not FDA approved, but recent publication shows 84% improved treatment potential in presence of these drugs. So we can, recently I have not provided data here, but we have also tested ivermectin in our lab in breast cancer cells which are hormone receptor positive. And we have shown that ivermectin is actually activating several pathways just like paclitaxel, which is an anti-cancer drug. Ivermectin, as you know, fenbendazole and mebendazole, they are anti-parasitic drugs. And some of them are like fenbendazole is a animal drug, deworming drug. Mebendazole is actually patented by Johns Hopkins, USA, but they are not letting this information widespread. This mebendazole is actually patented for cancers, and these also we want to use to functionalize our nanoparticles and see if there is a better efficacy, because these drugs have very, very low side effects as seen in hundreds of case files. And recently there is another drug, webdegestrant. As you can see, it is a drug approved by FDA by just last year for estrogen receptor positive breast cancers. This is showing better efficacy than several estrogen inhibitor drugs. So we would also like to explore these new drugs because they have not been worked upon in terms of hyperthermia. So as I previously mentioned, this is the overview of the in vivo evaluation in animal models. We will also test the biodistribution of the nanoparticles by ICP-MS and histology, which is usually done for animal experiments. And we will also assess the toxicity and safety assessment. We will carry out these experiments. And why we have designed this study, as I said, there is a limited efficiency of this nanocarrier mediated treatments in cancer, because many of these nanocarriers are accumulated in liver. So they have to some extent poor incompatibility. They are not really well tolerated. They tend to agglomerate in the body. They have insufficient magnetic response. And because of this, the simultaneous drug delivery, magnetic targeting, and hyperthermia treatment is also causing issues. So there is always a need for surface modification of these magnetic nanoparticles for better circulation and tumor uptake. So these are the overall conclusions of our work. Till now, we have successfully synthesized the nanoparticles at Dr. Kalyan Lakshmi's lab, and we have confirmed their crystalline phase formation by FTIR, XRD, and magnetic studies. But at present, we have successfully coated our nanoparticles with dextran, and now we are ready for drug loading and ligand attachment to carry out our in vitro studies. So finally, we would like to acknowledge our team, and especially Professor Brahmananda Manavati Garu, Project Director of Anrf-Pair, Central University, and also Professor Vijulata Mangadam, Principal Investigator and Project Coordinator, and the team at OU who has been helping us conduct interviews for selection of candidates for preparation of bills and submission, including non-UGC section. We are really thankful to everyone. And of course, our Honorable Vice Chancellor, Registrar Sir, and OSD Sir for helping us participate in this.
Thank you, Ma, for your wonderful presentation. And I thank each and everyone for helping us in conducting the webinar. Without your support and collaboration, this wouldn't have happened. And it is a very successful webinar as far as it is concerned. Almost hub and spokes.
I got disconnected. So that is our work, madam. Thanks a lot for giving us the opportunity to present.
Yeah. So I thank all the co-PIs for supporting this webinar. And I thank the Hub Institute and Professor Brahmananda Manavati, Project Director, Anrf-Pair HCU, and also Anrf for supporting our research. And amongst all the hub and
Madam,
spoke,
your audio is a bit breaking in the middle.
amongst all the hub and spokes institutes, we are the first one to conduct the webinar. And I once again thank all the co-PIs for your support and cooperation and for
Congratulations to you.
the timely presentation
Thank you, madam.
and scheduling according to the time schedule given to you all. Thank you very much.
Thank you and congratulations, Vijilata ma'am, for
Thank you,
conducting
thank you, ma'am.
here
Thank you, ma'am.
to make us do it first.
Thank you, ma'am.
Thank you, madam. Thank you, Vijilata madam.
Thank you, ma'am. Thank you.
Thank you, ma'am. It went very well, and I appreciate that you have finished in within such a short duration of time so much. And I hope I congratulate you and wish you all the best, ma'am.
And this went on with the Telangana state initiative of Pragati Palana and Praja Palana and Pragati Pranalika. And Professor Sirisha must be very happy with this
I'm
initiative.
eagerly waiting for today's report also.
Yeah, sure. I'll upload. I'll send it.
Yeah, yes,
Thank you.
ma'am.
Thank you.
Thank you.
Thank you, madam. Bye.
Yeah, bye.
Bye, bye.